So on Thursday last week, we were working our way through the immune system. We talked about the first line of defense, which included the physical and chemical barriers on the outside, like the skin, chemicals like, oil and saliva and, vaginal secretions, that kind of stuff. And we talked about part of the second lines of defense. We got through the antimicrobial substances, and I'm hopefully correct here, this is what we're ready to talk about. Two different types of cells, our natural killer cells and our phagocytes. Remember, this is all components of what we call the non-specific immune system, also known as our innate immune system, which is responsible for attacking things without kind of a memory component. So attacking fungi and viruses and bacteria the same, because they don't really recognize them differently. Okay. Now, last week and in your last unit, we talked about lymphocytes. We learned that there were 3 types. And what we'll talk a lot about here in just a little bit when we talk about adaptive immunity are our T cells and our B cells. Okay, T lymphocytes and B lymphocytes are 2 out of our 3. The third are the natural killer cells, which I always say, I wish they were called natural killer lymphocytes. About 5 to 10% of the lymphocytes in our blood, also present in places like our spleen, lymph nodes, and bone marrow. And as it states there, they kill non-specifically infected body cells and cancer cells. Okay, non-specifically, remember, no target in mind as far as recognition, that kind of stuff. When they bind to target cells, like what's shown down here at the bottom with this infected body cell, they release granules containing toxic substances that those natural killer cells have, which includes perforin, that's kind of shown here in red, a protein that inserts into the plasma membrane of a target cell and creates channels or perforations in the membrane. Perforations is just kind of a fancy word for holes. Okay, so it's like it pops a hole in the the infected body cell, in this case, causing extracellular fluid to flow into the target cell, which will cause the cell to burst. Cytolysis is a fancy word for that. So it's like we fill up a water balloon too much and it pops, right? That's what's going to take place here. Other granules include granzymes, shown here in yellow, which kind of use those holes that our perforations or our perforin proteins make, allowing the granzymes in. These are protein digesting enzymes, inducing target cell to undergo apoptosis, which is a fun word to say. It means the cell's going to be programmed to die. This type of attack kills infected cells, but not the microbes inside the cells. So the released microbes will be released into the extracellular fluid, but they may or may not be intact. Ultimately, they're going to be destroyed by our phagocytes. I don't know why I have 2 periods there. Phagocytes, remember, are specialized cells performing phagocytosis. Ingestion of microbes or other particles such as cellular debris. We'll talk about the 5 steps of phagocytosis here on the next page. The 2 major types, this is something I kind of pointed out in our blood lecture. Neutrophils and macrophages. As we know, macrophages are enlarged forms of the cells originally called monocytes. Single macrophage can engulf up to 100 bacteria during its lifespan, whereas a single neutrophil can engulf up to only about 20 bacteria before it dies. So macrophages are a little bit more, capable of digesting, breaking things down. In addition to being an innate defense mechanism, phagocytosis plays a vital role in our adaptive immunity, as we'll see here in a little bit. Okay, let's talk about those 5 stages of phagocytosis. Begins with something that I talked a lot about in our last unit when I talked about blood. Chemotaxis is, remember, that chemical attraction of white blood cells, in this case phagocytes, to areas of trauma like infection. Chemicals that attract phagocytes might come from the invading microbes themselves, might come from other white blood cells, damaged tissue cells also release chemicals, or even proteins of the complement system, which we talked about just a little bit the other day. Once that white blood cell, that phagocyte, has gotten to the area of trauma where the microbes are, then it adheres to it. Adhere means to like stick. In this case, what happens is, it's kind of cool, the plasma membrane of the phagocyte kind of extends itself, creating these extensions called pseudopods around where the, in this case, microbe is sticking to the cell. And then what happens is those pseudopods end up, it's like a bear hug. It hugs and pulls that substance in, creating a phagosome. Okay, so that's the step 3, ingestion. Plasma membrane of phagocytes extends projections called pseudopods, which engulf the microbe. Microbe is enclosed in a sac called a phagosome, which then is going to be kind of attacked by our lysosomes internally. Lysosomes, remember, are the garbage disposals of our cells. Those two things will fuse together, forming what's called a phagolysosome, where those enzymes that are in that lysosome now can go and attack the microbe or whatever it is that was ingested. So lysosomal enzymes can break things down like cell walls, degrade carbs, proteins, lipids, and nucleic acids. We've talked all about that before. Phagocytes also form lethal oxidants, causing a process called oxidative burst. And within about 30 minutes, most bacteria would be killed by those lysosomal enzymes. And then what's left over is undigestible material. We call this residual body that can be discharged from the cell, which is step 5 of the killing. It's really step 5 to me. Killing is kind of taking place in digestion. But whatever, I don't name the stages. I just present them to you guys. Now, very closely tied in with phagocytosis are the next, or is the next, non-specific defense mechanism, what we call inflammation. This is our next second line of defense. And it takes place in response to any type of tissue damage. And it's a non-specific defense mechanism, meaning that there is no difference in how we respond to things like cuts, to burns, to radiation, or bacterial or viral invasions. So things that produce inflammation, pathogens, abrasions, chemical irritations, distortion or disturbance of cells, extreme heat, extreme cold. So any kind of tissue damage, essentially. 4 major signs and symptoms of inflammation include redness, pain, heat, swelling, which are, you know, for the most part, if you think about like something like simple like a zit, okay, a zit is a little bit raised, so there's a little bit of swelling there. You touch it, a lot of times it's pretty painful, okay, and obviously it's a lot of times very red at first, and then, you know, it whites out or whatever. What's kind of missing there would be, you know, the heat. You know, a zit so small, you're not gonna be able to measure heat. If you have some extreme tissue damage, you can actually measure the amount of heat that is being produced. Also, sometimes you see possible loss of function. You know, think like a sprained ankle or something like that. A lot of that has to do with like pain and that kind of stuff that causes, you know, the joint not to work or whatever it is. Now, inflammation a lot of times is thought of as a bad thing, but it is a process that our body is doing something on purpose, and that is what I'm talking about right here, which I would highlight. The whole purpose of inflammation is an attempt to dispose of microbes, toxins, or foreign material at the site of injury, prevent their spread to other tissues, and to prepare the site for tissue repair in an attempt to restore tissue homeostasis. So it's not bad to have swelling, to have pain, all those things that go with it. You know, our body's doing something on purpose. What you don't really want is inflammation lingering. They call that chronic inflammation. That can be, you know, detrimental to your health. Acute inflammation, like when you sprain an ankle and your ankle swells up, those things aren't, bad by any means. Not to what a lot of people think anyway. 3 stages of the inflammatory response include vasodilation and increased permeability of blood vessels. We'll talk about that on the next page. And then phagocyte immigration, which we've actually talked a little bit about, which is where the phagocytes move from our blood into the interstitial fluid so they can clean things up. And then finally, tissue repair. Okay, let's talk about the first of the 3 steps or stages, whatever you want to call it. Vasodilation and increased permeability. Now, we haven't really talked a whole lot about vasodilation yet. We'll talk more about that in our last lecture of this unit. But vasodilation is where a blood vessel expands and opens up more, which allows more blood flow to go through it. When we talk about the term permeability, it means that substances normally retained in our blood are actually permitted to pass from our blood vessels out into the interstitial space. Okay? So we're going to increase blood flow to the area of damage and then let stuff leave the blood to enter the interstitial space. Okay? What the purpose is, vasodilation allows more blood to flow through the damaged area, and the increased permeability permits defensive proteins such as antibodies and clotting factors to enter the injured area from our blood. So it's basically going to allow things that can fix the damage to come there. Okay? When a person has a car wreck, the police come, fire trucks come, ambulance comes. It's kind of what's happening here. Okay? The fixer-uppers, people that are going to clean up the damage, are coming to the area of issue. Okay? So the increased blood flow also helps remove microbial toxins and dead cells. Among substances that contribute to vasodilation, increased permeability, and other aspects of inflammation include histamine, we've mentioned a little bit about before, released in response to injury by mast cells found in our connective tissue, basophils, as well as platelets, which we've talked about before. Neutrophils and macrophages are attracted to the site of injury, also stimulate the release of histamine, causing vasodilation and increased permeability. So histamine promotes both the vasodilation and the increased permeability produced by a lot of different cells. Kinins induce vasodilation and permeability and also serve as chemotactic agents, promoting chemotaxis, attracting those phagocytes like we just talked about. Prostaglandins released by damaged cells also help intensify the effects of histamine and kinins. They also stimulate the emigration of phagocytes, which we'll get into on the next page. Leukotrienes produced by basophils and mast cells increase permeability, permitting those defensive proteins and antibodies and clotting factors to enter the injured area from our blood. Already talked about that up there. They also aid in adherence of phagocytes to our pathogens so that they can then ingest them. We just talked about the 5 steps. Adherence was step 2. And then different components of the complement system, those different proteins stimulating the release of histamine, which we said all about what that does. Also, they attract neutrophils by chemotaxis, promote phagocytosis, and some components also destroy bacteria. Dilation of arterioles and increased permeability of capillaries produce 3 of our signs and symptoms of inflammation. Heat, because as we've talked about, blood contains heat in it, and if there's more blood in that area, there's more heat. Redness, which is due to the blood that's in the blood vessels. Fancy term for redness of the skin would be called erythema. And swelling, which is fancily known as edema. Okay, and the swelling, a lot of that's because we're allowing water and proteins out of the blood and into that damaged area in that interstitial space. So heat and redness results from the large amount of blood accumulating in the damaged area. As local temperature rises slightly, metabolic reactions proceed more rapidly and release additional heat. Edema or swelling results from the increased permeability of the blood vessels, permitting more fluid to move from our plasma into the spaces between the cells. Pain is another prime symptom of inflammation, as we just talked about. Results from injury to neurons and from toxic chemicals released by microbes. Kinins, a lot of times, like bradykinin, we've talked about that before, how that stimulates the sensation of pain. So there's, your 4 signs and symptoms of inflammation: heat, pain, swelling, and redness. Okay, our next stage of inflammation is the immigration of our phagocytes, we've talked about before, where those white blood cells have the ability to stick to the inner lining of our blood vessels and squeeze through those endothelial cells. Within an hour after the inflammatory process starts, phagocytes move from our bloodstream into the interstitial fluid to gather at the site of injury so that they can attack things like the microbes that might have got in when you have a paper cut through your skin. Okay. Immigration of our phagocytes involves 3 steps. The first is margination, where the phagocytes stick to the lining of the blood vessels. Then immigration, or diapedesis, as it's also known as, is where they squeeze between the endothelial cells to leave the blood. And then the final step is where they're going to actually go directly to the area of trauma. That's the chemotaxis, that chemical attraction, like me, being attracted to our kitchen in my house when my wife is baking brownies or something like that. Any chemical that attracts a phagocyte is called a chemoattractant. Talked about those on the previous page, or and previously as well. Phagocytes then can destroy the microbes as well as dead and damaged cells. Within a few days, a pocket of dead phagocytes and damaged tissue forms. This collection of dead cells and fluid is called pus, which is usually occurring in most inflammatory responses, usually until the infection subsides. Once again, going back to the example of a zit, you feel that zit, is it white-headed that same day? Typically not, right? Okay, typically it takes a day or 2 before you see the whitehead. The whitehead is that pus kind of leaking out, kind of crusting up. When you squeeze, you shoot the pus out. We've all probably popped a zit and know what that's like. Gross, I won't get into any more of it. Okay. Tissue repair is what takes place last. Once the infection has been contained and phagocytes have successfully killed the invading microbes, we can then repair the injured tissue. In this case, what we need to do is fix the skin that's been broken through. Looks like the epidermis has been clean cut through all the way down into the dermis. So we're going to have cell division occurring where we eventually connect the skin back together. What can actually happen a lot of times when you have tissue injury is formation of a scar. In this case, we probably wouldn't see one, but in like a super deep cut, really bad burn, scars form. I won't really get into scars, but that is fairly common. We all have them in various places. During tissue repair, worn out, damaged, or dead cells are replaced. Okay, and our final non-specific defense mechanism is fever. Something we experience when we're sick. One of the things tested, very commonly tested right now, a lot of schools and buildings you can't even go into without having your temperature checked. Easy way to see if something's wrong with the body. It's described as an abnormally high body temperature occurring because the hypothalamic thermostat has been reset. Remember learning about the hypothalamus, we mentioned that it's our thermostat, like the thermostat in your house, apartment, building, whatever you live in, where you set it at 70 degrees or whatever. If it drops below that, there are heating mechanisms that cause your house to heat up, like you kick your furnace on, then it heats it back up, trying to maintain it at 70, or when it's hot outside and it causes your house to warm up, air conditioning kicks on to cool it back down. We have our hypothalamic thermostat typically set around 37 degrees Celsius, but when a person has a fever, basically it's like walking over to your thermostat and hitting the up button. Okay, so cranking it up from 37 to 39 whatever, which occurs commonly during infection and inflammation because of what we call pyrogens, okay, which are mediators of inflammation that act on the hypothalamus to raise our body temperature's set point. So once again, just pushing the button up. Okay. Pyrogens can be exogenous, which means they come from outside the body, like bacteria, or endogenous, where they come from our own body cells. Exogenous outside, endogenous inside. Okay. Many bacterial toxins elevate body temperature, sometimes by triggering the release of fever-causing cytokines, like a chemical called interleukin-1 that our macrophages produce, histamine that we've mentioned a lot already from mast cells, and prostaglandins, which if I go back to this page, we talked about those as part of the inflammatory process. Okay. And what was one of our prime signs and symptoms of inflammation? Heat, right? Well, fever is, where you measure total body heat. Okay. Now, why do we do this? This bullet right here, I would highlight, start, whatever. Elevated body temperature intensifies the effects of our interferons. We talked about interferons last week. Inhibits the growth of some microbes and speed up body reactions that aid in repair. So it is a purposeful thing to try to help us out. People tend to think, oh man, my body has a fever, it's really bad, right? And then take, Tylenol, Advil, whatever, try to get rid of it. Okay, but your body's actually doing it on purpose to kind of enhance your immune system. Now, what you don't want is a super high fever. That's very dangerous. Okay. At the onset of a temperature, or really I should replace that word with a fever. At the onset of a fever, an elevated set point will cause our hypothalamus to induce sympathetic signals, causing vasoconstriction of our peripheral blood vessels in the skin in an attempt to prevent heat loss. We talked about that the other day, how we, when we're cold, we shift the blood away from our skin to our core. If this does not help conserve heat loss, we shiver. This is where the chills come from that people get. When our tissues heal, they stop releasing those various cytokines, those different chemicals, and our set point returns back to normal, which triggers our body to try to cool off. What do we do when we get hot? To cool off, we sweat, right? We also vasodilate the skin, and this explains why a lot of times if you wake up after a nap and your fever has broken, you wake up all wet, right? You've sweated it out, as they say. Your fever is broken, you've cooled off back to normal. I talked about, and I just said, people like to take, Tylenol, Advil, that kind of stuff. Aspirin, something we've mentioned before, back when we talked about it being a blood thinner. Not that it's truly a blood thinner and that it makes your blood more watery, but that it reduces the likelihood to clot because it works to stop the synthesis and release of prostaglandins. It's also an antipyretic because it reduces our fevers. It's also an analgesic, which means it's a pain reliever. It's also described as anti-inflammatory. Anybody ever seen a listing like on a bottle NSAID before, or when talking about different medicines? So it's NSAID. That's a class of drugs. NSAID stands for non-steroidal anti-inflammatory drug, and aspirin is one of those. So. Okay, table 22.1 just summarizes all those different first line, second lines of defense that make up our innate immune system, innate or non-specific, which unfortunately are not impenetrable, which means we might have to call in the big guns. Our adaptive immune system or our specific immune system, which provides us defense against specific invading agents, things like bacteria, toxins, viruses, and foreign tissues. These things that we recognize as foreign and provoke immune responses we call antigens. I've mentioned that a lot here in the last couple lectures. Immune system that performs our adaptive immunity includes cells and tissues that carry out the immune responses. Two properties distinguishing adaptive immunity from innate: specificity, meaning that we are recognizing particular foreign molecules called antigens again. That also involves distinguishing self from non-self molecules. In memory, we remember previously encountered antigens, which is great because a second encounter prompts an even more rapid and vigorous response, as we'll talk and see here in just a couple slides. This is where those T and B cells come into play. T lymphocytes, B lymphocytes, which we learned develop immunocompetence. For our B cells, it's in the bone marrow. For our T cells, it's in the thymus. Immunocompetence, remember I kind of described this as like a soldier going off to basic training and trained to be a killing machine, a soldier ready to carry out, in this case, adaptive immune responses. What's really taking place is that the B and T cells make several distinctive proteins that are inserted into their plasma membrane. Some of those act as antigen receptors, molecules able to recognize the specific antigens. Okay, there's two different types of adaptive immune responses: cell-mediated and antibody-mediated. And they're produced, or performed, I should say, by two different cells. T cells perform cell-mediated, B's produce our antibody-mediated immunity. Okay, T cells are once again derived from our thymus, and that they develop in our thymus. They aren't produced in our thymus. All blood cells, remember, are produced in our bone marrow, red bone marrow specifically. T cells are much more prominent than our B cells. 70-80% of our circulating lymphocytes in our blood, also found in places like our lymph nodes, spleen, and other lymphoid tissues like the appendix and our tonsils. Before leaving the thymus, they develop that immunocompetence, like we've mentioned a lot so far. And once they've matured, typically they exit as one of these two types of T cells, although there are two others that I want to talk about. Helper Ts or killer Ts are the two kind of most prominent major types. Now, there's other terms for them. Killer, kind of fancier term for killer T cells, are cytotoxic T cells. Cyto means cell, toxic, in this case it means toxic to the cell, killing the cell. I usually call them helper Ts or killer Ts. Another term for helper Ts are CD4, and CD8 is another term for killer Ts. The CD4 and CD8 come from the surface proteins on their plasma membrane. The stuff I won't really get into, it's all biochemical, pretty complicated stuff. But they do exactly what their name implies. Helper Ts help activate T lymphocytes or B lymphocytes to become plasma cells. So helper Ts help Ts and Bs. Okay. Killer Ts combine with the antigen on the surface of the foreign cell, causing lysis of the foreign cells and the release of cytokines, chemical messengers of our immune system that we've talked about as recently as a couple slides ago. Memory Ts remain inactive until future exposure to that same antigen occurs. If they encounter that same antigen at that later time, they respond by a swift reaction because there are thousands of them. And regulatory T cells are going to provide a break, kind of in parentheses here, or quotes I should say, on the specific immune system or immune response, meaning that they kind of slow down our immune response by inhibiting the killer T cells and B cells, which is great because it prevents diseases caused by our immune system, things like allergies and autoimmune diseases, multiple sclerosis and lupus, and things we'll talk a little bit about towards the end of the lecture. So essentially what they do is prevent our immune system from attacking normal body antigens, although they're not 100% effective. We all suffer from some forms of allergies. You know, I'm not going to get into any of that, but we all know what we're allergic to. B cells are produced in our bone marrow and produce our antibody-mediated immunity. They also not only are produced in our bone marrow, but develop and become immunocompetent in our bone marrow, where they can leave and become 20 to 30% of our circulating lymphocytes in our blood, but also places like the lymph nodes, spleen, and other lymphoid tissues. When they become sensitized to an antigen, they proliferate and differentiate to form clones of daughter cells that either produce antibodies against that antigen, that's what our plasma cells do, or become memory B cells. I'm going to kind of do something to kind of show you what I mean. So bear with me here real quick. So we start with our B cells, which can either produce memory B cells or plasma cells. Plasma cells are the cells that produce our antibodies. So drawing some lines here. B cells can ultimately produce one or the other here, memory Bs or plasma cells. The immune response is a result of the production of antibodies, which are the proteins that are produced by our plasma cells. Now you might be asking yourself, what's the function of memory B cells? Well, memory B cells can transform and become plasma cells. Now, I'm being really loose with things here, but this is essentially what can happen. And I'm going to do something kind of funky here. But memory B cells can reproduce themselves. So that's me drawing a circular arrow as best I can. So memory B cells can produce more memory B cells. That way we can produce more plasma cells to produce more antibodies. So that's my kind of flow chart of what's going on. I'll talk a little bit more about each of those cells here in a minute, but first let's talk more about cell-mediated immunity, which once again involves those T cells, not the B cells. Cell-mediated immunity, as it says there, is particularly effective against intracellular pathogens, which means that are things inside of cells, like viruses that have already made their way into and infected a cell. Remember, intra means within. They're also effective against cancer cells and foreign tissue transplants. Begins with the activation of a small number of T cells by a specific antigen. Clone cells are then formed, which recognize the same antigen as the original lymphocyte. Some of these clone cells become effector cells, while others become the memory cells. Effector cells carry out the immune responses that ultimately result in elimination of the intruder. This includes the helper Ts, which are part of the helper T cell clone, and active cytotoxic or killer T cells, which are part of the cytotoxic or killer T cell clone. So ultimately, what we have is just a whole bunch of cells being involved, not only for cell-mediated, but we'll also see the same thing with antibody-mediated immunity.
We don't have that last sentence. Should we write it down? It just stops at an intruder on my paper, at least. Do you guys have it?
Don't worry about it. Okay. I must have added it later. It's not something I'm going to ask you to. Sorry about that.
That's okay. I got confused. I was like, am I on the right page?
Sometimes I add stuff and then forget to add it to the student version. Now, cell-mediated, we said, was performed by T cells and was effective against intracellular pathogens. As you see here on this page, antibody-mediated immunity is effective against extracellular pathogens, which means that are outside of our cells, which includes viruses, bacteria, or fungi, not inside of cells, but in body fluids. Another term for body fluids are body humors, which is why this is sometimes referred as humoral immunity, because it involves antibodies attacking things in our humors, our body fluids. Okay. This is where we use the antibodies, obviously, since it's antibody-mediated immunity. Remember, we just kind of drew it all out. It begins with B cells transforming into plasma cells that then produce the antibodies. We can describe plasma cells as the protein factories, or excuse me, antibody factories of our body. Okay. Proteins is what an antibody are that circulate in our body fluids and our blood. Okay. Each B cell produces only one specific type of antibody protein. In other words, an antibody that can bind only to that one specific antigen. The other cells that are formed when we expose our antigens to our B cells would be the memory B cells to go along with those plasma cells. Along with that, we produce a lot more B cells as well. Plasma cells, once again, are the factories for producing antibody proteins. A few days after exposure to an antigen, a plasma cell secretes hundreds of millions of antibodies for about 4 to 5 days before it dies. Memory cells, and this is memory B cells here, do not actively participate in the immune response to the antigen. Instead, they quickly proliferate or divide and differentiate into more plasma cells and more memory B cells should that same antigen reappear at a future time. Down at the bottom, with some asterisks, kind of help you kind of really pay attention to it. Something I will talk more about on the next page, but that it's important to understand that these two different forms of immunity are not really working independently of one another, which is demonstrated that our helper Ts, which, as we said, T cells perform cell-mediated. Helper T cells do that, but they also, as it states here, help with antibody-mediated immunity, which we'll show in this portion of our figure here on the next page. So reading through, in most cases, when a particular antigen initially enters our body, there's only a small group of lymphocytes, the helper Ts, the killer Ts, and the B cells that have the correct antigen receptors to respond to that antigen. Depending on its location, a given antigen can provoke both types of adaptive immune responses due to the fact that when a specific antigen invades the body, usually there's more copy or many copies of it spread throughout. Some of those will be found in our body fluids, invoking an antibody-mediated response. Some of them will make their way into cells, infecting them, which results in a cell-mediated immune response. So some copies of antigen may be present inside body cells, provoking a cell-mediated response. Other copies will be in the extracellular fluid, provoking an antibody-mediated immune response. This helps illustrate, and so does the figure over here, that cell-mediated, antibody-mediated immune responses often work together. There's another example of things working together in our body. We call that integration to eliminate a large number of copies of our particular antigen in our body. Figure on the right is showing you the T cells and B cells involved. Remember, T cells originate in our bone marrow, but then mature in our thymus. Two major types are the killer Ts and the helpers, which the killers do the actual killing and destroying of things like the intracellular pathogens, cancer cells, and tissue transplants. Helper Ts help do that, but they also help those B cells form our plasma cells, which are the ones that produce the antibodies, or the memory B cells, which, as we talked about, can produce eventually future plasma cells. And then the antibodies are the things that actually work against the viruses and other things outside of our body cells. Speaking of antibodies, we're going to spend a few slides talking about them here on this page, next page, and maybe one or two more after that. Another term for antibodies are immunoglobulins. We learned about that in our blood lecture because they are part of the gamma globulin class and are immune in function. They are produced, as I've said a lot already today, by plasma cells that form our B lymphocytes. There's kind of some pretty crazy shapes and features to our antibodies, but they consist at least of 2 short or light chains and 2 long or heavy chains, as well as antigen binding sites that are highly specific for only one particular antigen. And the binding of that antigen to its specific antibody produces or results in production of more antibodies that are specific to that antigen. Here's kind of an illustration that shows the 5 different classes and what their kind of shape is. A lot of them have this kind of capital Y shape to them. IgA is kind of a double Y. IgMs always look to me like a snowflake, and then the Ds and the E's look just like the G's at the top. Okay. The Ig that you see in all of them are short for immunoglobulins. So immunoglobulin M, A, E, G, and immunoglobulin E are the 5 classes. I've listed off a little bit about each class, which I do want you to know. The first antibody class to be secreted by plasma cells after initial exposure to an antigen are the IgMs. They also activate the complement system, those proteins that act like a domino effect or produce kind of a domino effect, causing agglutination and lysis of microbes. Remember, agglutination is something we've talked about when we talked about blood typing. We mentioned that agglutination with regards to blood is a bad thing, like what happens when you receive a wrong blood transfusion. Agglutination is where things clump. We talked about that. Basically, the same thing occurs here with microbes. Lysis just means they die. Essentially, they kind of break down. IgMs also serve as antigen receptors. IgAs are found mainly in our mucous membranes, places like our nose, our saliva, our tears, bronchial, so that being your windpipe, and well, not so much the windpipe, that's your trachea, but your tubes in your lungs, the intestinal and vaginal secretions as well. We all know what those are, but block the entrances of those antigens into the body. IgDs help activate other B cells. IgGs are the most abundant class, protecting us against bacteria and viruses by enhancing phagocytosis, neutralizing toxins, triggering the complement system. Something else I would highlight, that they are the only antibodies that can cross the placenta from mom to fetus, which the placenta we haven't talked about yet in this class is the connection between mom and baby, allowing for nutrients to be given to baby when they're inside the womb, because babies aren't eating and drinking and breathing oxygen in. They rely on mom to get it, and the placenta connects to the umbilical cord that then connects to the baby. So IgGs are a way to, or IgGs are actually transferred over to baby before they're even born, which is really unique and cool. And then IgEs involved with allergies, as well as aiding in immunity against parasitic worms and other parasites. Now, those 5 different classes of antibodies differ in some of their actions, but all of them act to disable antigens in some way. And we've got them listed here on this page and illustrated on this page. The first is just neutralizing antigens. Reaction of an antibody with an antigen blocks or neutralizes some bacterial toxins and prevents attachment of some viruses to body cells. So essentially, in this case, the toxin is being neutralized because the Y-shaped antibody is bound to it. Letter B there on the right is showing you what happens when microbes become agglutinated. Agglutination, again, think of as clumping, and I even say in parentheses there. So because antibodies have 2 or more sites for binding to antigen, the antigen-antibody reaction may cross-link pathogens to one another. And we can see that here. They're connected to one another, which basically creates a cluster or a clump of them. Okay, it's like chaining them up together. It'd be like taking a bunch of criminals and handcuffing them to one another. It's hard for them to all work at the same time if they're trying to get away, and it'd be easier to track them down. In this case, clumping together allows for phagocytes to ingest them more easily. And that's kind of a theme for a lot of what our antibodies do. They're not necessarily going to do the killing themselves, but they just make it easier for something else. A lot of times it's a phagocyte to do the killing. They precipitate antigens. When you think of precipitation, what do you think of? You know, like rain or snow, something coming, you know, down in the form of liquid. Okay. Antibodies can cross-link soluble antigens, so things that are dissolved into complexes that are too large to stay in solution, so they get pulled out of the fluid. As a result, the complex precipitates out of solution, making it easier for the antigen to be phagocytized. They help activate the complement system, specifically the classical pathway. There's also a non-classical pathway. Basically, it's another way to, you know, activate those dominoes to fall down. And then the final one is opsonization, where antibodies can function as opsonins that coat the surface of a microbe, thereby making it easier for phagocytes to engulf and destroy it. So, like I said, antibodies really don't necessarily do the killing. They just kind of help enhance it and make it easier for something else to do the killing. All right, that table just kind of lists off a lot of the same stuff I already talked about. Not something I'm going to ask you to know. I just, kind of one reason I just included some kind of cool pictures of them. Okay, last thing I really want to talk specifically about for antibodies are what we call the primary and secondary responses. When a person is exposed to a particular pathogen for the first time, there is a latent. I've seen and said that word latent a lot this semester. Just think of latent, and it might not help, or it might not hurt, I should say. Think latent, think delayed. You can write it down that way if you want to. So if you replace the word latent with delayed, there's a delayed period of 5 to 10 days. So about a week before we see measurable amounts of antibodies appearing in the blood, at least measurable amounts. So you have like a week that goes by where maybe the virus is in your body and you're not producing antibodies against it. And in this time, what we call the primary response, it might not be quick enough to keep you from getting sick. But look what happens when you get a subsequent second exposure to it, triggering a secondary response where we produce antibodies much more rapidly, so much more rapidly that we see maximum amounts of antibodies occurring within 2 hours. 2 hours is a heck of a lot faster than 5 to 10 days. And hopefully this should be quick enough to prevent you from acquiring the disease. And it's this immunological memory that provides the basis for acquiring immunity against pathogens, demonstrates why vaccinations are such a great, amazing thing, because vaccinations help you have a secondary response instead of a primary response, whatever that vaccination was against, whether it be, you know, the chicken pox or the measles or the mumps or whatever, or hopefully the coronavirus here before too much longer. I lied to you. We're not quite done talking about antibodies because I want to talk about how we acquire them. How do we get adaptive immunity? Kind of 4 different things that go into it. First 2 to go together, whether we acquire it actively or passively, which refers to if we produce it ourselves or not. Okay. Produce it, I'm talking about antibodies. Actively, we're producing our own antibodies and immune system cells, so those T's, those B's, those plasma cells, etc., against the specific antigen. Whereas passively, it's where we receive antibodies from another person or animal. So that would be, you know, not our own body, because there's no memory component involved. Immunity passively acquired is only temporary, lasts until those antibodies degrade. Adaptive immunity can also be acquired naturally, which means that we are exposing ourselves to the antigen by chance, a natural event, like someone sneezes on us, okay? Whereas artificially, meaning that we develop the immunity as a result of artificial means, like deliberate exposure to an antigen through vaccination. Now, I used the term, or I talked about chickenpox. I just mentioned that a little bit ago. I am a little bit older than you guys, some of you more than others. I had the chickenpox as a kid, but you guys are probably at the generation where some of you had the chickenpox and maybe more of you did not have the chickenpox because there was a vaccination that was developed about that time when you guys were really little. When I was young and before I was even around, they used to have what they called chickenpox parties. Anybody ever heard of this? Anybody ever not heard of this? Chickenpox party is exactly what it sounds like. It was common for like one parent on the block or one family on the block that, you know, a kid in that family got the chickenpox, and there was a agreed upon thing between all the other families. If your kid has it, call my kid up and we'll bring them over so that we can expose them to the chickenpox. They get the chickenpox, they can be done with it when they're little, because chickenpox is, you know, an older person, can be really dangerous actually. It's a lot more deadly than when you get older. So that's kind of a artificial / natural thing where it's truly natural because you're you know, developing it by chance, but you're deliberately doing it. So it's kind of in that gray area. But based on all these factors, here are the 4 methods in which you can acquire adaptive immunity. Active natural is, once again, you're going to produce it yourself. Okay, and natural refers to the fact that you are exposed by chance. Okay, active artificial, meaning you're producing your own, but it's artificial, so it's through a vaccination. Okay, whereas passive natural means that you're transferred antibodies, like in the case from mom to baby across the placenta, or actually IgA antibodies can be given to baby through breastfeeding. Another great example of why breastfeeding is such a great, wonderful thing, along with being free, because formula is expensive. And then passive artificial is where you can obtain antibodies intravenously from another human or animal donor. Okay, artificial is, you know, you're receiving a vaccination basically, but passively you're not producing your own. So the difference really relates with when you get, you know, like a chickenpox vaccine or something like that. A lot of times what you're getting is like a weakened or dead version of a virus, and then your body produces its own antibodies. But with this last one, you can basically just receive those antibodies. But remember, it's not going to be a long-term solution because eventually those are going to kind of die off. I do want you to know this table. This is kind of one of the few times in the semester where I do ask you to know a table. This is one of them. Okay, so we've been talking about how wonderful and amazing our immune system is. Unfortunately, our immune system can kind of suck at the same time. Not so much as it doesn't work good enough to keep us from getting sick, but that it almost works too well and fails to display what we call self-tolerance, which means that it attacks its own body's tissues. Examples of various autoimmune diseases include multiple sclerosis, MS. Now I could go into a lot more detail for all of these, and I've just really, really briefly given you an outline on what those different diseases are. In MS, the white matter of the brain and spinal cord are destroyed. So very, very, obviously impairing to normal body function. Myasthenia gravis is where there's an impaired communication between nerves and skeletal muscles. Type 1 diabetes, we've mentioned before, is where the beta cells of the pancreas are destroyed, resulting in an inability to produce insulin. Rheumatoid arthritis destroys joints, and really that's kind of a very, very broad way of saying it. When you think of arthritis, who do you think of? Old people, right? That's osteoarthritis. That's what happens like when our joints just get, old and brittle. The cartilages are kind of gone. The joints start to hurt. Rheumatoid arthritis has not really aged anything to do with it. In fact, a lot of people that suffer from rheumatoid arthritis are kids, which is just kind of sad to think about. Graves' disease, antibody-induced stimulation of the thyroid gland. We talked about that a little bit in our endocrine lecture. And then lupus is where the body's immune system attacks its own tissues and organs, including joints, skin, body, or it can be kidneys, blood cells, brain, heart, lungs. So it's kind of a whole body autoimmune disorder. Therapies and treatments for various autoimmune diseases include removal of the thymus gland, injection of beta interferon, immunosuppressive drugs, and kind of a really cool process called plasmapheresis, where the person's blood plasma is filtered, removing antibodies and antibody complexes. So it basically is kind of a cleaning of the blood, getting rid of some of those antibodies. And then something we all suffer from, as I've already said, allergies. This time of year might be a time of year where people are really suffering from them as well. Allergies involve a person being overly reactive to a substance that is tolerated by most other people. Another term for allergic is hypersensitive. The things that induce an allergic reaction are called allergens. Common ones would be certain foods like milk, peanuts, shellfish, eggs, various antibiotics like penicillin, tetracycline, venoms like from bees, spiders, snakes, cosmetics, poison ivy, pollen dust, and mold. Two different types of allergies, immediate and delayed. Exactly what it sounds like, whether the response is pretty much right then and there, or sometimes within a day or 2 to 3 for delayed. For whatever reason, immediate involves abnormal B cell response, whereas delayed would be an abnormal T cell response. You might think of anything that you might, come across, and then like 2 or 3 days later, you might, experience that allergic response, like itchiness, redness, possibly. How about that one right there? Poison ivy is a good example of one that a lot of people have delayed hypersensitivity to. And not everybody's susceptible to poison ivy. In fact, really, I don't know if I am. I've walked through it before, and I'm pretty lucky in that I've never really had a breakout. But other people know, like my cousin, if he just sees it, he's gonna suffer from it. Okay, regardless if it's delayed or immediate, it involves those antibodies of the IgE class. Okay, I got an illustration here that I'll explain with this big paragraph. In response to the first exposure to certain allergens, some people produce IgE antibodies that bind to receptors on the surface of our mast cells and our basophils. This process is called sensitization. So here's the first exposure. Oops, wrong way. Okay. The next time that same allergen enters the body, it attaches to the IgE antibodies already present. In response, our mast cells and basophils undergo degranulation, releasing histamine and preformed granules. After degranulation occurs, mast cells and basophils also release leukotrienes and prostaglandins, all these chemicals we've mentioned here, especially when we talked about inflammation. Collectively, they function as inflammatory agents, dilating our blood vessels, which is why the area might appear red, increase blood capillary permeability, so causing swelling, contract smooth muscles in the airways of the lungs, why you might feel wheezy, increase mucus secretion, why your nose might run, and stimulate nerve endings that cause pain, and obviously a lot of times things can be kind of itchy. Okay. So it's the chemicals that our own body cells are producing that really is what's causing the allergic reaction, itchy skin, dry, watery eyes, runny nose, that kind of stuff that we all experience because of those IgE antibodies. Now, what's a type of drug that a person takes to try to prevent allergies? Antihistamines, okay? Because as we just mentioned, histamine does a lot of those things like make you sneeze and make your nose run and that kind of stuff. Last thing I want to talk about is HIV and AIDS, because there's a relationship between HIV, which is a virus, and what it does to our immune system, ultimately causing AIDS. Okay, AIDS is actually an abbreviation, if you were not familiar with that. Now you know. Short for Acquired Immunodeficiency Syndrome, where a person experiences telltale assortment of infections due to progressive destruction of the immune system cells by the HIV virus. HIV is short for human immunodeficiency virus, a virus that destroys those helper T cells that we've talked about today. When a person is given the HIV virus through whatever means it is, a lot of times sexually, those HIV viruses, I talked about this before, but they like to invade host cells and tell that host cell to then make more copies of the virus. Kind of come overriding the function of that original cell. When a helper T cell is invaded, look at the number. Over 10 billion viral copies may be produced each day. And what ends up happening is that original virus ends up killing that host cell and then releasing a whole bunch of more viruses that can go and infect another host cell, eventually just knocking off T cell after T cell after T cell, which does what to your immune system? It impairs it, okay? By the end stage of infection by HIV, the person develops AIDS, where their immune system is so shot that they're susceptible to what we call opportunistic diseases or opportunistic infections, which are things like a common cold that a normal person fights off easily, but a person with AIDS cannot. When a person dies of AIDS, they die because they catch something like a cold and they just it kills them. You don't die of HIV. You don't die of that virus. Die because your immune system sucks at this point.
So wait, it does like the opposite of what antigens do. Well, wait, whatever the ones are that don't kill off diseases but just make it easier.
Yeah, essentially yes. Yeah, so they, unfortunately, now there's a lot of important drugs, like they call it the cocktail. It's a whole host of different drugs, which is why, like Magic Johnson was diagnosed with it when we were like in the early 90s, late 80s, and he's still alive. Unfortunately, it costs a heck of a lot of money. Places like Africa where HIV and AIDS is really prominent, they don't have the same access to those same drugs, unfortunately. It's a lot more deadly for them. Okay, any questions on anything immune system related?